Hello everyone, I'm Libby Jones and I'm a researcher at the Institute of Marine Research in Tromsø, Norway. I'd like to thank you for the opportunity to talk to you today about ocean acidification dynamics in the marginal ice zone of the Barents Sea. I would like to thank my co-authors and numerous people that have helped with this work. This is a contribution to the Nansen Legacy project. The Barents Sea is located in the Atlantic part of the Arctic Ocean. Atlantic waters flow north in the West Spitsbergen Current and enter the Barents Sea in the south, but also via boundary currents along the northern Svalbard shelf. Arctic waters flow south into the region and act as a transport pathway for sea ice to enter the Barents Sea. The region has experienced warming, loss of sea ice, and increased Atlantic water influence or Atlantification over the last decades. In order to investigate the impact of these processes on the oceanography and ecosystem, the Nansen Legacy project has carried out a series of seasonal transects in the northwestern part of the Barents Sea. Today I will present data from late summer in August and winter in December along the transect where a series of process stations or P stations were carried out. These ice charts show the sea ice extent and main ice types in relation to our transect in the northwestern Barents Sea. At the beginning of August, the summer ice edge was located between P4 and P5 at about 80 degrees north. In December, the sea ice cover had extended southwards and the winter ice edge was forming between P2 and P3 at about 78 degrees north. The distribution of temperature in the full water column can be used to identify the key water masses in the region. The warm and salty Atlantic water can be seen dominating the full water column in the south, but also as subsurface boundary currents in the north. In August, less sea ice cover and seasonal warming created a larger proportion of the warm polar water in the upper layers. In December, cooling, freezing, and southward extension of the sea ice created a greater proportion of the cold, fresh polar water in the upper water column. The sea ice cover, coupled to upper ocean physics and seasonal processes, strongly influence the distribution of dissolved inorganic carbon. In August, primary production and biological carbon uptake reduced the concentration of dissolved inorganic carbon in the surface layer. Meltwater inputs from the retreating sea ice also act to reduce the concentration of dissolved inorganic carbon. In December, vertical mixing, remineralisation, and brine rejection from the forming sea ice enable the water column to become enriched in dissolved inorganic carbon. Similarly to the dissolved inorganic carbon, the presence of different water masses strongly influences the total alkalinity of seawater, otherwise known as the buffer capacity. These panels show the temperature and salinity space with respect to alkalinity for August on the left and December on the right. The Atlantic water is warm and salty and characterized by high alkalinity. In August, the greater influence of sea ice melt and meltwater inputs shows a larger proportion of the fresher polar water, characterized by lower alkalinity. By December, the reduced meltwater influence returns the water column to more salty conditions from the Atlantic water with higher alkalinity. The cycling of inorganic nutrients with inorganic carbon revealed some distinct seasonality. The plots show nitrate and phosphate and inorganic carbon and nitrates in August in the top panels and December in the lower panels. In August, strong biological uptake had occurred in the meltwater influenced surface waters. Higher carbon to nitrate uptake ratios were the result of strong nitrate drawdown reaching depletion levels following the growing season phytoplankton blooms. In December, remineralisation increased the concentrations of the macronutrients and the inorganic carbon in the water column. Gradients in the upper water column can be used to determine key processes that drive changes in the inorganic carbon and alkalinity. The plots here show the vertical distribution of inorganic carbon in the upper 400 metres, where we see a gradient between 120 metres depth and the surface. Here we can look at the total change and split this into key driving processes, such as those resulting from salinity changes, those resulting from primary production and remineralisation, changes due to the formation and dissolution of calcium carbonate, and finally, residual changes on inorganic carbon due to air-sea CO2 fluxes. These plots show the changes in alkalinity in the upper part of the water column, in August in the top panel and December in the bottom panel along the transect. The largest changes occurred in August, which were strongly driven by meltwater dilution that created the largest alkalinity deficits, particularly in the ice-influenced waters. The impact of biological production had a small effect on alkalinity, contributing to slight increases due to nitrate uptake. Calcium carbonate processes showed that mainly alkalinity is removed from the water column during summer, and this is due to the formation of calcium carbonate shells. One exception was noticed at P6 in the Atlantic water influence region, where an increase in alkalinity could result from the dissolution of calcium carbonate, either from calcium carbonate shells or from calcium carbonate minerals released from melting sea ice. This plot shows the changes in inorganic carbon, again in August in the top panel and December in the bottom panel. Similarly to alkalinity, the larger changes occurred during August, but the distributions in December showed a smaller but similar imprint from those in summer. Primary production drives the deficits in inorganic carbon in both the ice-free or the low ice-covered waters. Changes in salinity also had a notable effect, particularly in August due to the release of the meltwater from the retreating sea ice. The effects on calcium carbonate on the DIC were very small, but the contributions from CO2 fluxes indicate a stronger effect in December and likely result from CO2 uptake from the atmosphere or a contribution of CO2 in the saline brines that are rejected from the sea ice. From the distributions in alkalinity and dissolved inorganic carbon, we can determine the calcium carbonate saturation state for aragonite, and we use this as an indicator of the state of ocean acidification. So these plots show the distribution of the aragonite saturated state in the full water column, in August on the left and December on the right. You can see the impacts of the primary production and meltwater dilution that yields high saturation states in the upper part of the water column. The Atlantic water influence at P1 in the south and at P6 and P7 in the north shows the inputs of alkalinity from the Atlantic water that helps to elevate saturation states. In contrast, in the more Arctic and fresh water influence, for example at P2, we see that the saturation states are low during both seasons. In the deep Nansen Basin in the north, the aragonite saturation horizon is located at about 2,000 metres. During December, we had the opportunity to investigate the distribution and abundance of calcifiers in the water column. The plot on the right shows the different depth intervals and size classes of pteropods, the green bars, along the transect. Pteropods had very low abundance or were nearly absent in the Atlantic water influence stations, for example, P1, P6 and P7. For the organisms that were found there, the subpolar species Limacina retroversa was dominant. In contrast, in the more Arctic-like conditions of the central Barents Sea, the highest pteropod abundances occurred, of which the polar species Limacina helicina was dominant. These results have indicated that the different pteropod species have separate ecological preferences, perhaps strongly influenced by the water masses and the chemistry. This work is currently in revision in Frontiers in Marine Science. To take a closer look at the pteropod abundance in relation to the water column chemistry, to show the distribution of aragonite saturation states in the upper 400 meters. I've just highlighted the conditions that were found at P2, which was typical Arctic-like conditions, which showed seasonally low saturation states. We can see here that the values for the aragonite saturation in the subsurface layer was actually less than the critical values for Limacina helicina and may present ocean acidification conditions and be detrimental to shell formation. The work carried out within the Nansen Legacy project is helping to better resolve seasonal variability in the Barents Sea, where future warming, sea ice loss, and greater Atlantic water influence could increase the nutrient supply and lengthen the productive season to drive enhanced biological carbon uptake. Coupled to inputs of alkalinity from the Atlantic water, these processes would counteract dilution effects and buffer against acidification in the Barents Sea. And with that, I'd like to thank you all for your attention.